Hi, welcome to ClinChem 1. We're going to do Lab Essentials and Safety. This is chapters 1 and 2 of the Donna Larson textbook. Okay, so first of all, let's talk about clinical laboratories. So it's been said to produce anywhere from 70 to 80% of objective data that doctors use to, and nurses use obviously to diagnose and treat patients, monitor patients, etc. So we do generate a lot of data that medical decisions are made on, so we're definitely an important and integral part of the healthcare team. So clinical labs can be found in hospitals. There's a lot of employment in hospital labs. They're busy. They're open, 24/7, and there's always depending on the size of the hospital, they can be there's always work to do. It's always busy. There are some also in outpatient clinics and physicians' offices, and so these obviously would have more what we call normal hours, even though there's urgent care hours and stuff like that. So just because a tech works in a physician's office lab, it doesn't mean that they don't work maybe weekends or evening hours and stuff like that. Then of course we have reference laboratories. These are huge laboratories. The nearest one to our location is probably going to be AEL, American Esoteric Laboratories, over in Memphis, Tennessee. And there, of course, there are state and federal labs. They're usually part of if they're state, usually part of the state health department. So for here in Arkansas, the Arkansas Department of Health has a lab. That's where a lot of the newborn screenings and all that are done. And these are often, an example also is the CDC, but the state and federal labs often report to the CDC. CDC is obviously a federal entity. Okay, so a little bit on regulation and accreditation of laboratories. So the regulation of all laboratories falls under the CLIA, so the Clinical Laboratory Improvement Act of '88. They call it CLIA '88, even though they've done some amendments and added. We just kind of still go with that original date, and so everybody knows what they're talking about. And so basically, the aim of CLIA '88 is to establish standards of quality that all labs in the United States have to abide by. And then sometimes you'll see stuff like if a test is labeled a WAVE test or high complexity test, and all, those are things that have been established by CLIA, and it also dictates on who's allowed to run that test. Anyway, so all labs that are open in operation have to fall under the regulation or are regulated by CLIA '88. Then labs can be accredited also. So accreditation is usually voluntary. So the Joint Commission is one of the accrediting bodies, but usually they come in to look. It's for hospital labs. They do a lot of hospital accreditations and stuff, and then the big one for the labs is going to be CAP accredited. So CAP takes things a step beyond CLIA '88, and it's like a stamp, like a high quality stamp, if you will, of approval on a lab. It takes a lot of work to be CAP accredited and then to maintain the accreditation. And to maintain CAP accreditation, a lab has to do proficiency testing and competency testing and document such when they are inspected by CAP. Proficiency testing, it happens when the CAP sends, if you will, like kits with unknowns. Usually there's like at least 5. They're label 1, 2, 3, 4, 5, or A, B, C, D, E samples, and they are usually for a specific department or a specific type of test or tests, plural. And they're sent for, it's usually for a bench, if you will. So like for chemistry, they will send you one, and then you would have to run all the tests listed. And so it's probably going to be all the tests that the lab performs, and they have to be run, and you have to report out the results on those unknowns. You don't know what they're supposed to be. So you test them and you report exactly the values, just like if it's like a fake patient, if you will. Or a mock patient, and you send all your results back, and they compare your results to what it should be and also to what everybody else got that got the same samples, and then send your report back, and you either, pass some or fail some. Hopefully you pass all of them. But if maybe your machine wasn't working just right and there's one or two tests that aren't accurate, then you'll get dinged on that, and you have to fix it so that you can pass the next one. If you fail 3 proficiency tests from CAP on a specific analyte in a row, then they can take away your ability to get reimbursed for that test, and basically, essentially, to offer that test. And so it's a really important thing. So this allows labs to make sure that they are turning out accurate tests. And then competency testing has to do with your techs knowing how to run, all the benches that they're assigned to. So if a tech is supposed to be working, for example, in chemistry and in hematology, they have a checklist when they're first trained of all the items and the things they need to be able to know how to do, and then you need to test them on that, or at least go back through the checklist yearly to make sure that it can still do everything. And then it's good sometimes to do refreshers on things maybe that aren't done very often, maybe some body fluid testing and different things like that, where to make sure they stay sharp and know how to handle those types of samples. Okay, so your first poll question is going to be, any Abacus clinic lab, for example, at a minimum would have to be CLIA certified, CAP accredited, or both? What do you think? Okay, let's talk about the different materials that we find in the lab. So first of all, you can have glassware and plasticware. And those things can be like, some beakers and stuff of water. Plasticware can be anything from like pipettes, test tubes. This is actually a urinalysis tube, test tubes for blood, containers or plastic containers and stuff like that. They will have centrifuges also. I have one of those in my office. And they will have balances, pipettes, some automated. So this is not automated. This is a micropipette. It's another micropipette. We're going to talk about those. And then also like serologic and volumetric pipettes. And those are like these types of things here. And then there's reagents. Reagents that are used in the lab have to go through FDA approvals. Everything, testing, test kits, reagents, all of that is regulated by the FDA. You can't just launch something in the market without having FDA approval. And then the water that's used in the lab has to be reagent grade. We're going to talk about the different types of water in the lab. So let's dive a little deeper into the lab glassware. So borosilicate is used a lot. This is Pyrex. And so like this is a pretty typical one. This is my beaker of water I was walking around with, and I don't know if y'all can see if I can get it to focus, but maybe I'll move it back. But it says actually Pyrex right here. A lot of your cookware and glassware that you use in the kitchen is usually Pyrex. So anyway, Pyrex is often used for volumetric type stuff, so you're measuring volume of water and stuff, and liquids and etc. Corex is 6 times stronger than borosilicate or Pyrex. It is strengthened chemically and it will resist clouding and scratching, which means it's more expensive, and it's a higher quality glass, stronger. And then low actinic is amber or red colored for light sensitive materials. I don't have one in here, but this photo right here represent a lot of your quality control vials are that way, especially if they contain any kind of analyte that would be degraded by light. So there's dark color vials are often used in the lab. Okay, so let's talk a little bit about plasticware. So first type of plastic is polypropylene. Your plastic pipette tips, some specimen and test tubes are polypropylene, and so I know here it is. So we have, for example, a pipette tip like that, and then this is polypropylene, this type of plastic stuff. So polyethylene, certain test tubes, bottles, graduated tubes, disposable transfer pipettes, which I have here, the squishy kind, those are polyethylene. You have to be careful with some of these. They can sometimes bioabsorbed dyes, it sticks to it, or stains, and then some proteins sometimes can bind into this. So if you suck up like a urine sample or something like that, I know like THC will bind to some of these, and so if you use it to load your a cup or whatever, if it binds in here, then it's no longer going to be in your sample. Anyway. Then you have polycarbonate, certain graduated cylinders and flasks. They're usually pretty clear. I've got a picture of them here coming up, but like this would be, an example. It's a graduated cylinder, so pretty straight up there. So that's polycarbonate, and then polystyrene is usually rigid and clear, but it will splinter, and it can be capped with graduated tubes and test tubes or that way. A good one, a good example is a Cova tube for your analysis. If you break this or step on this or find a broken one in your box, it will be in splinters. You have to be careful not to cut yourself on it and stuff. And then you also have Teflon. I don't have Teflon here, but the advantage of Teflon is that it's chemically inert. It has a wide temperature range. It is often used in stirring bars, but tubing and bottle cap liners. So tubing is where you're going to see a lot of it in chemistry. So pretty much any tubing that is used to move reagents and to move samples and stuff like that is going to be Teflon. So here are the pictures as promised. So again, polypropylene, the pipette tips, polyethylene, the disposable transfer pipettes, an example, polycarbonate. So this is the type of plastic vials and containers that are polycarbonate. Polystyrene, like the Cova tube, it can splinter very easily, and then Teflon, chemically inert, doesn't react, and it has a wide temperature range, and things don't stick to it, so pretty cool. So let's talk a little bit about centrifuges. So there are several types of centrifuges you can see in the lab. There's the swinging bucket centrifuge. Usually this is a pretty big countertop type centrifuge with a huge lid. This is what's portrayed right here, and there's buckets that are swinging like that, and you have tube inserts for various size tubes. And when the centrifuge starts spinning and it's spinning, those buckets actually go like this. They go horizontal, and then they're spinning that way, really, 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 really fast, obviously. And then that's what, of course, causes the centrifugal action. And then you have fixed angle rotors. These are more like that, and so these are usually smaller centrifuge, and when you load the tubes, they're usually loaded at an angle all the way around, and they stay at that angle the entire time they're spinning. Ultra centrifuges are big centrifuges that are floor models. They often have swinging buckets. They have really high speed, and then some of those can also be refrigerated. So sometimes if there's a specific type of testing that requires that the specimen stays, at a refrigerated temperature, but also has to spin for like 15 or 20 minutes or some really long time, where it would risk warming up if it was in a regular centrifuge, and you can use a refrigerated centrifuge. Again, this is the looking down into the swinging buckets, but it could be also an ultra centrifuge, big floor model one. So the relative centrifugal force is the force times gravity, or G-force. It is a force that is required to separate two phases. And so in blood, we have the solid phases, the cells, right? And we want to separate that from the plasma or from the serum, depending if you have clotted blood. And then the solid is your blood clot. And so the centrifuge will put the solid phase stuff at the bottom, the cells at the bottom, and then the liquid portion at the top, the plasma or the serum. RPMs are revolutions per minute, and it is how fast the centrifuge is spinning, and there are different requirements for different types of testing, and so that's usually established in the protocols. And so oftentimes in lab, they could have centrifuges set for different types of specimen, depending on how fast. There's not a lot of variety on how fast you have to run it, and it's pretty easy to find settings that accommodate the most samples. The RCF is the relative centrifugal force. It is calculated by multiplying, so 1.1118 times 10 to the negative fifth times the radius times RPM squared, and that gives you the relative centrifugal force. Centrifuges, basically, you need to do maintenance and calibration on them, so there's a way to calibrate it. When you look at a centrifuge, you can look in the lab. On the lid is usually a hole or something you can see. In the middle of the rotor, there's usually like a line, or or it's half black, half white, and you can see it spinning. So you can put the calibrator on top of it, and then you can set it to send, let's say, spin at 5,000 RPM, and then the calibrator will read to see exactly how fast it is spinning. Is it spinning at 5,000, or is it 4,900, or is it 5,100, or whatever? So mostly the maintenance that has to be done on centrifuges is wiping them down if there's any kind of spills of blood or serum or plasma, or any yuckies or broken tubes that are shattered and are all over. You need to clean it up and wipe it down on at least a daily basis, but it's good to wipe and clean the centrifuges. So how do you load a centrifuge for it to be balanced? So centrifuges need to be balanced. If a centrifuge is not balanced, it can literally like walk off the counter, and so especially if it's a counter centrifuge. So what happens is it's off balance, and it goes like that, like a washer that's off balance, and then it moves, and then it can fall off the counter. We don't want to do that. So there are several things to consider in loading a centrifuge, making sure that it is balanced. So first is where you put the tubes, and so I'm showing you here some loading patterns. So usually you just want to find the center point, the middle, and you want things to be balanced on both sides. So you can simply load them equally across like this. Here, where they're equally across, this is pretty easy right there. But then you're like, this is all good for all even numbers, right? Well, what if you have an odd number? What if you have only 3? Well, a lot of times there's a way to balance it 3 ways. And there's even a way to balance 5. If you look at the spacing here, right, that is actually balanced. You can even do 7 this way. You're basically trying to spread the weight out evenly all the way around the centrifuge. And you can do 9. So you pretty much do anyway. The only one you can't do is just 1. You have to have a balanced tube. So there's a couple other things to consider. When these patterns are assuming that each of the tube is equally full and it's the same size tube. So you have, tubes that are like this that hold like 6-7 mils or something like that. You have some bigger, fatter ones that hold 10 mils, and then you have some that are like about this big that are shorter that hold more like 4 or 5 mils and stuff like that. So you can't balance one of these big ones with a short one, that they're not equal weight and stuff. So you want to balance the same type of tubes that have the same type of volume in them. And so a lot of times in the labs, what they'll do is they have balanced tubes. So they're tubes like these that have water. So you'll have maybe one that's half full, one that's a quarter full, and all that. And you can adjust, you can add or remove some of the water to match it to your specimen, and then you can use that as a counterbalance tube in your centrifuge to make sure that your centrifuge is balanced. Okay, so in this little exercise in the Nearpod, I want you to mark how we load 5 samples. So this is a draw it activity. This is our first one for the semester, and so you can actually select here the little drawing tool, and you can select your color. Like you could do red, and then you can draw and stuff. So anyway, I'm gonna undo this, or just leave it like that. It's fine. So anyway, so do that for 5, and on this one, do 3 different ways to load 6 samples. And then in this one, you want to load 10 samples in a balanced way. So again, you're just going to use a bright color that can be seen, but you can hide this out of your way by clicking up here, and then you can go start noting where you would put your 10 samples in a way that is balanced. So this is a swinging bucket. Again, just be mindful of where the middle is, and try to draw lines to connect your samples and stuff. Okay, so there's also usually some balances, although they're not used heavily in clinical labs. They're still there because you may have to measure something or weigh a specimen or sample or something like that. So there are several types of balances. You have an equal arm substitution balance right here, and then this is a magnetic force restoration balance, which is also a top loading balance. Obviously, you can just say set something on top of there and it will read it. And then it's electronic because it has an electronic digital display and stuff. And the advantage of some of the electronic ones is you can convert between, you know, metric and imperial. So you can have it in grams, or you can have it in ounces, or in pounds and ounces, or whatever. And so you can tear it and you can do all kinds of fun stuff to it. Okay, so let's talk a little bit about volumetric labware, how to measure volume. So our main instrument that we use to do that is pipettes. All right, so we're going to first talk about the different types of pipettes that are in existence. So there's to deliver and to contain. The majority of the pipettes that you're going to find in a lab are going to be to deliver. And so the to deliver, drain by gravity, you want to hold them straight. And three of the examples are the serological, and the volumetric. And so, for example, so this is a volumetric. The volumetric one has a bulb, and you see this one has a number 5, and it has a line at the top and a bulb, and it goes, it's a tip. There's no graduation up and down. Okay, and so that's a volumetric. The 5 means it will hold 5 mL. So if you fill it up, so the line here, the bottom of the meniscus is at the bottom of the line here, you have exactly 5 mL. You cannot use this for anything other than to deliver 5 mL of the liquid that is in there. Okay, and what it means is once you sucked it up to, you know, got the liquid all the way up to the meniscus, and then you put it in your container there to deliver the water or whatever you had in there, once you hold it straight and everything has been drained all the way down, it has delivered 5 mL, even if you see little drops of water still inside. It has delivered 5 mL. Okay, so that's the volumetric one. There's also the serological. So the serological ones you probably have seen, they have numbers, graduation all the way up and down. And so you can, this one is a 5 mL serological, so you could, for example, only use it for 3 mL or something like that, or 2.5. You can measure, you know, their various different volumes. Okay, and then so you have the difference between, there's a more and just a serological. So the more ones are not graduated to the tip. So the one I was showing you, the 5 mL one, it's not graduated to the tip. So the last line, there's just the last line, and they're generally meant to be full. You fill it all the way up, and then you just dispense down from that in a controlled fashion, and you know, and then whatever you've delivered, that's how much has been delivered. In a serological one, there are lines like all the way to the tip. So that's the difference between more and serological ones. And then the to deliver blowout is usually a serological pipette, and it has 2 bands on the end. And so like my little 5 mL, it's a blowout when you can see the 2 lines right there. Whereas this other one that I had, the serological, that's not the more. This one's the more, the 5 mL. This one is also 5 mL, but it's just serological. There's not a double band on that end. So the blowout means that after it has all the way drained, you want to blow some air through, and so usually that's done by putting a bulb and then, you know, squeezing some air. This one has a little cotton ball, so if you actually push it, it'll move. Yeah, the cotton ball. There you go. And so you need to blow it out for it to deliver everything. So the other one I'm skipping over is the to contain. It's called Solly one. It's like one of these ones. You have to rinse it out, and then that allows it to get everything out. These were used to be used for like hemoglobin measurements and all that. I have never used a to contain one in the lab. They're pretty much all to deliver, and the majority of the disposable ones that we use in the lab are to deliver blowout ones. And then we have all of our micropipettes. So there are two types. There are air displacement type of pipettes, and it's basically a piston and a plastic pipette tip attached to it. So these are all, air displacement type of pipettes. And so what's pretty typical about an air displacement pipette is, micropipette, there's one of them, is like you have a piston that you engage here, and there's we'll find there's a first stop, second stop, and some of them are adjustable. So this one's one where you can adjust it, right, up to a specific volume, and whatever you've adjusted it to, when you press down to the first stop, it pushes that volume of air out, and then usually you have to attach a pipette tip to it, right? And so you press down, it is displaced the setting. So let's say it's 100 microliters, it has displaced 100 microliters of air out, and then you put it in a liquid, you can see it, and then you release, and it will suck up that set volume, okay? And so that is, most pipettes are air displacement type of things. And they're air displacement because, I don't know if you're going to be able to see this or not, I'm trying to do. If I did this underwater, when I press down, there's bubbles. I make bubbles, okay? You really don't want to do that in your specimen. You usually want to press down, and then press down first, and then put it in the water, and then release, and then you have your volume, whatever your volume was set to on that you set here. Okay, there's another there too that is in your picture, and it's this one. So this is also an air displacement one. The difference is this one's not adjustable. For example, this one is meant to contain 200 microliters. That's the only volume it can contain, and there's not a first stop, second stop. It's just you press all the way down. Same thing. You put the pipette tip. It's displacing air also. And you press it down, put it in the liquid straight up, and then release it, and it'll suck up 200 microliter of the liquid. And this is because that's the setting that it's on, not adjustable, but pretty consistent. So those are the two types. And then there's positive displacement pipettes. And so they have a capillary tip usually, and the liquid is drawn in and then pushed out by squeegee action. This is often used in a transfer of liquids. It is reusable, and there's a wash or flush step in between samples. You will see these in the analyzers, moving fluids back and forth and stuff like that. Think of it as a syringe, right, where you would suck liquid up, right, the squeegee, and then push it back out. And so it would go into the liquid and then suck it up, and then go somewhere else, whatever, and then dispense it. Okay, so that it's slightly different there than the air displacement. So you have the sucking it into the syringe or into a tubing or something like that. So that's positive displacement. You also have to do pipette calibrations. Usually there's one person in the lab that's assigned to do that, and this is to make sure that the volume that is said, for example, the 200 microliter, is what it's actually delivering in pipetting and stuff. Okay, so now if you just pipetted 5 mils of clinical lab reagent water, that's what CLRW stands for, into a QC vial, using a disposable 10 mil serological pipette with one band around the top, what do you do next? So you need to select one of these answers. That's your activity. And then just to see if you can remember everything we talked about, this is a little matching activity. And so, for example, click on this, and then what type of centrifuge you think it is, and you look at the options, and then if you get it right, both you click on it, but click on the like this can be fixed angle, and it'll turn either green or it'll turn red. So work through that activity. We are going to talk about water purification. So CLRW, as I just stated, is the abbreviation for Clinical Lab Reagent Water. So it is really important to have good, pure water in the lab, and this is because we use it to reconstitute calibrators, to reconstitute quality control. It's used in the machines to reconstitute different reagents and stuff like that, washing, and so many things. You cannot just use tap water, because if you think about the process that water goes through to become tap water, a lot of times it's chemically treated and has other things. So the water that comes out of the tap has, some chlorine and some other things in there. So there are several methods to create clinical lab reagent water. We have reverse osmosis. And reverse osmosis is water is forced through a semi-permeable membrane. And this is more of a molecular filter. So just water that's come out of a reverse osmosis filter is not clinical lab reagent water, but it's a good first step. So it removes the bigger stuff, the bigger molecules and stuff like that, the contaminants and bacteria and different things like that. So reverse osmosis is a good first step. And then it can go through distillation or deionization. So in distillation, water is vaporized and then recondensed. Again, distillation alone does not produce clinical lab reagent water. It does produce, however, distilled water. And so you should also, it needs filter, and it also needs a carbon or ion exchange type of filtering process to make clinical lab reagent water. And then deionization is running the water through ion exchange filters. It removes certain ions using either anion or cation exchange resins, and it ends up being a mineral-free deionized water. So distilled, deionized, there are differences there, and so pay close attention to requirements. And so if it's okay to just use distilled water, it will say that, but generally you want clinical lab reagent water. And there are water systems, like the Millipore is one of them, that combines all of that, and then that's your clean water source, and it is connected to the analyzer that needs it, and it feeds clinical lab reagent water to the analyzer, and is usually a port or something that you can get clinical lab reagent water from the Millipore directly into a container or whatever, and then you can, a clean container obviously, and then you can use it to reconstitute your quality control or whatever you need to do. To monitor water purification, we look at resistivity. So it's the measure of the electrical resistance, and it will assess ionic content. So if you have a high ion content, you will have low resistivity. So basically what it is it's running electricity through the water. The more ions there are, the easier it is for the electricity to jump across the ions and to pass through the water. The least ions there are, the more it's just water molecules, the less little jump points it has, and the harder it is for us to cross across a path of water within a water cell. So for clinical lab reagent water, you want the resistivity to be greater than 10 milliohms per centimeter, and you also want to check bacterial content. So I have something that's pretty kind of related. It is a measure of resistivity. It's just what it gives, the measurement it gives you is like, is it total dissolved solids measurement thing. And so my water purification system has one of these. So you can turn it on and it'll say 000, and then if you put it in, and here I have tap water, it will give you a reading. So the reading here is 1.22. I don't know if you can see it. And so that is not very pure, but there's it changes if I move it. Okay, now it's saying 0.96. Anyway, it's saying 0.96. Anyway, so that's the number of parts per million here in this water, and so basically the more the parts per million, the lower the resistivity, and the easiest it is for the current to pass around, and basically the more contaminated the water is, the more things it has. And so again, it can be maybe it's not a bad thing. So for example, if you bought some spring water, some mineral water that had a high magnesium content, which is really good for your health, it would have a higher parts per million reading because it's reading all the magnesium and other minerals that are contained. But you wouldn't want to use that to make up reagents and stuff like that, because you'd be contaminating it with magnesium and other things that are contained in the water. So anyway, so that's a little bit on water. You do need to understand solution concentrations. So the molarity, or moles of solute per liter of solution, it is used to calculate the concentration of a solution, and it's the molecular weight times the molarity, gives you grams per liter. The normality is a gram of equivalents of solute per liter of solution, and percent solutions, which are often used, are parts of solute per 100 parts of solution. So the percent solutions, you will see sometimes you will be, if you're on the phone with tech and you're having to fix an analyzer or something and they want you to clean it or to run something through it, that they may say, hey, I need you to run a 10% bleach solution through. And so what it means exactly that, you need to calculate. It's pretty easy to do, but for example, if you had a beaker that holds 100 mils, or a container, a cup that holds 100 mils, let me see, this one holds 100 mils, right? Well, you can measure out 10 mils of bleach, that's 1 part, and 9 mils of, lab water. And so, 90 mils, sorry, 90 mils. So 10 mils of bleach, 90 mils of lab water, and you would have 100 mils total, and 10% of that is bleach, because 10 mils out of 100 mils is 10%, or 1/10. So the percent solutions are often used, and if you make one up, then you need to label it in a cup and put, say just 10% bleach, 25% bleach, or whatever, 5% bleach, or something else. Sometimes other cleaners that can be used. Enzyme activity is measured in international units. An international unit is the amount of enzymes that will catalyze a reaction of 1 micromole of substrate per minute, and I use a lot, again, for our enzymes. All right, let's talk dilutions. So dilutions are made in chemistry, usually because a certain analyte in a sample is too high to read. And so every analyte that we analyze in chemistry has a certain measurement range that the analyzer can measure. It can't measure below a certain amount, it can't measure above a certain amount. So it has a range in which it can operate. And if your concentration is beyond that range, just way more than what the analyzer can measure, then you may be required to dilute your sample. Now, granted, there's a lot of analyzers that automatically dilute that for you. So if it's too high, it'll automatically do a 1/2 dilution, a 1/3 dilution. So, but it's not always the case. And then sometimes even doing that, it gets beyond the range, and then you have to go back and do manual dilutions to be able to see if you can get a reading. And then some analyzers, you do have to manually dilute it and then program the dilution factors in the analyzer before you run it. Okay, so let's review this. So 1/2 dilution is made by mixing 1 part of the sample with 1 part of the diluent. So please look at the notation. So I prefer the fraction, and you'll see why, because to me it makes more sense. So a 1/2, when you write the fraction, is 1 part over the whole, which is 2 parts. Okay, so a 1/2 dilution, you have 1 part sample, 1 part of the diluent, so it's 1/1, so there are 2 parts total, and that's 1/2 is 1/2. Now, sometimes you will see in instructions, depending on where in the world they're coming from, they'll call it a 1-to-1 dilution, and it means 1 part sample, 1 part diluent, 1-to-1, which is essentially a 1/2. But you can see how this can get confusing, because then if you move here, you have a 1-to-2. Is a 1-to-2 a 1/2? No, 1-to-2 is a 1/3. And so it's good to just pick one thing and stick with it, and I prefer using the fractions. The reason is that let's say you have a glucose that is reading beyond the analytical range and you decide, I'm going to do a 1/2 dilution. So you mix 1 part of your plasma, 1 part of your diluent, put it in a little cup, load it back on the analyzer and run it. And now you get a glucose result. And the glucose result on that diluted sample is reading 500, let's say. Now it's a pretty round number, it's 500. Well, that's the diluted. The diluted is 500. So what's the patient result? Well, the patient is, if you dilute it by half, then you have to multiply it by 2. And so you would do 500 times 2 is 1,000. So your actual glucose concentration in your original sample is 1,000, the one you couldn't get a reading on. So what's in your patient is a blood glucose of 1,000. Okay. And so the reason I like the fraction is that bottom number is what you multiply your result back by to get the actual initial concentration in the sample. So the example of the 1/2, you would take 1 ml of serum and 1 ml of lab water, and that would be a total volume of 2 ml, thus making it a 1/2 dilution. So moving on to 1/3 dilution, you would mix 1 part of sample with 2 parts of diluent, so it's a 1:2, right? For a total volume of 3 parts, so you would do 1 ml of serum, 2 ml of water, total is 3 ml, it's a 1/3. And if you had done a 1/3 dilution and your glucose read 500 on your 1/3 dilution, then the glucose in your original sample would have been 1,500, okay, which is still possible. I've seen glucose that high. And then a 1/10 dilution is made by mixing 1 part of sample with 9 parts of diluent, so it's a 1:9 in the total volume of 10 parts, right? So 1 ml of serum, 9 ml of water, and you get 10 ml, just making it 1:10 or 1/10, sorry, a 1/10. And then on that one, you would multiply the results by 10. Now, you rarely would have to ever dilute a glucose to 1/10, but for example, you would have to dilute creatine kinase. These guys can run really, really high. So CK creatine kinase is the normal is usually less than 200 international units per liter. You can get readings up to 100,000. So you can, and usually most analyzers will read up to 1,000. So you may have to do some pretty heavy dilution if you have a really high reading on one of those. And I use mils here just to introduce you to the concept, but the reality is usually you're working with smaller volumes, so this might then become 100 microliters plus 100 microliters. 100 microliters of serum, 100 microliters of water, and something. So you have to be able to think in the units that you're using. Okay, so we're going to start with some problems you can answer. So if the dilution is written as 1 and then colon 7, the dilution factor is, think about it for a second, what we just talked about, and pick an answer. And next, so we're going to do a little problem. So you need to do a 1/5 dilution on any patient's sample. You can only use 100 microliters of sample. How much diluent or water will you use? So select an answer. And then this explains then the first problem. So let's hope that you selected the right answer. I want you to think through it first, right? So if you need to do 1/5 dilution on your patient's sample, you're limited to using 100 microliter a sample. How much water or diluent do you use? So if you start with 100 microliter a sample, 100 microliter times 1 part, right? So it's 100 microliter. Then your total volume, if you want to do a 1/5, it's going to be 500 microliters. So 100 times 5 parts, that would be all. So you need 100 microliters of your sample. You need 500 microliters total. So how much water is that? You will take the total that you need minus your sample, your 100 microliters, and that will give you 400 microliters. So the answer was 400 microliters. So 100 microliters of sample, 400 microliters of water put together, you have 500 microliters, and the sample has been diluted as in a 1/5. And then when your analysis is complete, you multiply the results by 5. Now, if you have to program your analyzer and it's asking you what the dilution factor is, in this case, the answer is 5. The dilution factor is 5, which means that once it's got its reading, it will multiply its reading by 5 and do the math for you, which is really nice. So that's another thing to pay attention to when you're messing with dilutions on an analyzer is if you programmed it, did you remember to program the dilution factor, and is it doing the math for you, or do you have to still do the math afterwards? So something to pay attention to. Okay, so this one you need to do a 1/3 dilution on your patient samples. You are limited to using 250 microliters a sample. How much diluent of water would you use? Same again, think through. 250 microliters, you need to do a 1/3. How much water are we using? And then on this one, also, once you get the results, what would you multiply of your diluted sample? What will you multiply the results by to get the original concentration? So pick here, and so let's work through this one. So you do need to do 1/3 dilution on your patient sample, limited to 250 microliters. How much water? So 250 of sample is your 1 part. Your total volume will be 250 times 3 parts because this is a 1/3, right? 3 parts total. So that's 750 total, and then your diluent volume then will be your total minus your sample, so 750 minus 250, and it would be 500 mils. So you take 250 of your sample, 500 of water, and together that's 750 microliters in this case, and it is a 1/3 dilution. And then you would multiply your diluted results by 3 to have your original concentration. And so let's say your 1/3 dilution on an outer range ALT is now reading 450 international units per liter. What is the actual ALT level in the sample, in the original sample? So do some little quick math in your head, shouldn't be too hard, and select an answer. And then we're going to move on to all the safety stuff. So OSHA is the Occupational Safety and Health Act. We obviously, anybody that's working falls under OSHA. The goal of OSHA is, it is a, laws, regulations and stuff like that, is to create safe workplaces, including hospitals and clinical labs, etc. There are a lot of regulations that you have to follow, and that the workplace has to follow to, fall under OSHA, follow the OSHA regulations and stuff like that. And a lot of times there might be some training. So some of the examples you can give for safe work environments would be, they may, for example, if you're a nurse or working on a regular nursing floor, they may train you how to use, the hospital needs to train you, obviously, how to use some of the lifts to lift patients. So, because if you have a 500-pound patient or a 700-pound patient, you could very easily injure yourself trying to move them, and so a lot of them are going to be moved using lifts and stuff, and so you have to know how to get the lift set up and all of that, and then how to operate it without harming the patient or harming yourself. And so this is just one example. There are so many things that can fall under OSHA, but basically the goal is to allow you to have a safe workplace. So then you have the occupational exposure to hazardous chemical in laboratory standard that labs have to abide by. So generally speaking, in the clinical lab, we only use small amounts of chemicals. Your reagent packs are not, they're not huge. We're not like, lugging gallons of stuff. Actually the things that we have in the highest volume that would be in gallons and 5 gallons and cubes, it's going to be saline in deionized water, pretty much. With any kind of chemical, you have to know what the route of exposure might be. And so that is something that you have at risk of inhaling. Is it, contact to the skin, something you don't want to ingest, or etc. You need to maintain a chemical inventory in the lab. So any and all chemicals that are present in the lab, there has to be an inventory list of them. And also where they're stored. So they need to be properly stored. For example, all your flammables have to be stored in a flammables cabinet and stuff. So things like that. Some of the, isopropyl alcohol, ethyl alcohol, and all of that kind of stuff. Some of them may be just good to be stored at room temperature on a shelf, but you have to store them properly. You also need to have information on how to clean up chemical spills. So that you don't make things worse, because you think, oh, I could add water. Sometimes adding water to a chemical can make it more reactive and stuff and cause gases to form or something like that. So you want to make sure that you know exactly how to clean them. And every lab needs a chemical hygiene plan, which then would have all of this stuff like how to store it, how to clean it up, what the risks are, etc., etc. And it should be specific to that lab. The Hazard Communication Standard, it protects workers from illness or injury. It's training about different chemical hazards and protective equipment to use to handle those chemicals. There are 4 steps that are necessary to educate and train lab workers. First, you have to have a written plan, which is going to be in your chemical hygiene plan and stuff like that. All chemicals have to be labeled. And so, like, literally, we're talking about making a 10% bleach solution. If you made a 10% bleach solution in this thing, and you were not going to just use it and dispose of it. So let's say this thing had a lid and you were going to store it for a little bit. It has to be labeled as to what it is and properly labeled. And there's some sticker labels and stuff that can be used. And of course, so any and all chemicals in the lab have to be labeled, including distilled water, deionized water, and all that kind of stuff. Because if you think about it, a clear liquid like this, which this is obviously just tap water, but you don't know. It could be isopropyl alcohol, it could be ethyl alcohol, it could be, be anything really that's clear. And so it needs to be labeled. Safety data sheets, or MSDS, material safety data sheets, have to be present in the lab for all chemicals. These will tell you, like if you were exposed, what kind of symptoms you might get, also how to clean up, how to store, like all of those things. Anything that you need to know about the chemical should be on that material safety data sheet, and it needs to be available to you if you're working with that chemical. And of course, there are training programs on how to properly work with the different hazards and chemicals and stuff like that. The bloodborne pathogen standards. So this one is definitely very applicable to the lab. The aim is to protect workers from blood and body fluid pathogens. So one of the things, like when I started the program, is you had to have your hepatitis B vaccine because it wasn't part of the childhood immunizations. This has now become part of the childhood immunizations, and most people have been vaccinated against hepatitis B, but it is one of those the implementation of that vaccine also was to protect workers, because if they got a needle stick, it was very easy for them to get hepatitis B or HIV. So obviously you need to wear appropriate personal protective equipment, gloves, lab coats, face shields, glasses, and stuff like that. Now we also have masks and stuff. The PPE usually has to be provided to you by your employer. And then there has to be an exposure control plan. So what are the protocols? What is in place to reduce your exposure to bloodborne pathogens? And are you following, of course, these protocols in plan? But they have to have a plan, like your employer has to have a plan in place to protect you. Okay. And so within the bloodborne pathogen standards, you need to understand the difference between universal precautions and standard precautions. So universal precautions basically pertain to blood and bloody fluid. So you can obviously with your naked eye see that this is blood. So you have a blood sample, that would be a universal precaution. And the idea is to treat any blood sample like they have everything that you don't want to get. Okay, like they're heavily contaminated and stuff. And so this also would pertain to any body fluid that has obvious blood in it. But then there are the standard precautions would fall for things that have no obvious blood but are still a body fluid. So this could be things such as saliva, could be things such as urine and all of that. But basically, they both want to, you need to be treating these like it could potentially be contaminated. And part of the bloodborne pathogen standards is to decontaminate your workbenches after each lab and stuff. And so that's going to be one of your responsibilities. Even if you can't see that there's any blood or anything, you still want to decontaminate them as part of, at the very least, standard precautions. your Needle Stick Safety and Prevention Act, and we're using now safer medical devices. So you will see there are a lot of needleless systems for infusions and stuff like that, wherein we're talking about like if a nurse has to like inject something within a line, they're no longer using needles now. There's usually a way to lock them into and then to push the medication in. For the lab, we have a lot of engineering controls for the needles. So they're self-sheathing needles. And so there's one here that's displayed where once you've got your sample, you can just push this down over the needle. But I would even say it's dangerous to push it down with your thumb, because if it misses you, then your thumb is headed straight for the needle tip. So I usually like better to flip it and use a bench or another surface to close it, not your hand and stuff. So you don't want your hands anywhere near the needles. There's also in the syringe we were using, also there's this one is self-sheathing. So like once you're done, you can actually pull this all the way and it locks out, and then it protects you from being stuck by the needle. You should never recap needles, because if you're trying to put the cap back on, you could stab yourself too. But all of this stuff, like when I started, it was the stuff did not exist. It was just needles, and you try to recap it, and you could get stuck and all of that. Of course, we also have the sharps disposal containers, which are puncture proof, that you want to dispose all of your sharps in, including so all your needles and stuff like that. And if there's any kind of sharps injury, so if you do get stuck by a needle or there's a defective device and it causes you injury and all of that, it has to be documented. And then usually they need to see, is this something where they maybe need to switch brands, the retraining on how to close it. Again, the example I just used was simple, so not closing the safety thing with your hand, but using something like a countertop or something else, or a bed or a chair or something to close it for yourself, and not so that your hands go nowhere near the needle. So sometimes some retraining of the individuals might need to be done so that they have safer protocols. All right, standard operating procedures, or SOPs. So standard operating procedures are in place to reduce the risk of exposure for lab techs and any lab personnel. The part of a standard operating procedure will include, and this is for like any and every test that is done in the lab, or procedure that is done in the lab has a standard operating procedure, and you should be able to access it and see it. And it will say things like, what reagents are needed, what standards are used, calibration, what controls, what equipment do you need to run this test, what analyzer it's run on, it's a detailed step of the procedure, any kind of hazardous reagents that's used. So anything and everything that you need to know on how to do that test should be in a standard operating procedure. You should be able to, having had no training, read through it, and of course know what the words mean, but and be able to follow that protocol and perform the test in a safe manner and accurately. And this is what basically standard operating procedures, or your labs that you're going to do, your lab procedures are basically your standard operating procedures for every test that we do. Okay, so if you hurt your back lifting a heavy cube of reagent water because it was stocked on the lowest shelf, which standard will protect you as an employee? So is it OSHA? Is it occupational exposure to hazardous chemicals? Is it the hazard communication standard or the bloodborne pathogen standard? Next, you get blood or serum splash in your face when you take the cap off of a tube. Can happen. Which standard then does this fall under? So is it the occupation exposure to hazardous chemicals in the lab? Is it the hazard communication standard? Is it the bloodborne pathogen standard, or the Needle Stick Safety and Prevention Act? Right. A little bit about the warning signs and labels that can be used in the lab. The NFPA labels right here are often used, and each component has a meaning. So red is for fire, the radioactivity is yellow, and then a health hazard is blue, and then other stuff, other details is going to be in white. And the numbers go from 0 to 4, with 4 being the highest rating. So that means that something that's 4 for red is highly flammable and could very well, explode or catch fire very easily. If it's a 3, it's, a significant health hazard if you're exposed to it, etc. We put biohazard signs on anything that can be contaminated or potentially contaminated. So this is to indicate that precaution needs to be taken. You should have, of course, eye wash stations in all the labs. We have them in our lab, and also like the shower, exposure showers. Refrigerators should have, if you are holding biohazards in there, should be labeled as such. And you have to be very careful. For example, you work in a lab, the refrigerator that you store all your reagents in is not where you put your lunch or your drink, or etc. You can't do that. So you should have a refrigerator in the break room for food items, and it's not the same that what you use for reagent. And of course, vice versa, you don't want to go be putting reagents into the break room refrigerator and stuff. It's a no-go. Lab coats also need to be worn in the lab and not out of the lab. There's strict regulations on that too, and exits and stuff need to be noted, so in case of an emergency evacuation. All right, so sulfuric acid's biggest threat, you think, is it a health, combustion, so a fire, reactivity, or mixing it with water? And you can click on it and you can see it. And so tell me what its biggest risk is, and pick one. There. So biohazardous waste disposal, so human blood and blood components, body fluids, microbiological waste, pathological waste, all has to be disposed of properly. So the sharps waste container must be sealed prior to disposal, and usually ideally only contains sharps. So you will see sharps containers and biohazard containers, and because a hospital has to pay, or the facility, in this case for us the university, has to pay to dispose of this, and it is expensive. You don't want to do things like put regular trash in the biohazard because it makes it heavier and there's more to pay for. You don't want to put things that are non-sharps in the sharps container, etc. You want to make sure that you dispose of things properly, and anything that is biohazard must be disposed of in a biohazard labeled container. Yes, these things get picked up and disposed of properly, and it costs money to do so. What if you have to ship biohazardous materials? So this is governed by IATA. There are specific packaging, labeling, and documentation that's necessary to ship any type of medical or biological samples. You have to be properly trained on how to package it, etc., and label it, and all of this kind of stuff. So this would apply if you were having to try to ship samples somewhere. A lot of times now that when we have reference labs, they have couriers that come and get the sample, so you're not having to ship it through the postal system or FedEx or stuff like that. But sometimes you do have to send stuff through the postal system, and so there's a specific way to do that. Laboratory safety equipment. So there are biological safety cabinets. There are different classes depending on how much protection is needed. There are so many different ways where these things can work. Some of them have a specific airflow pattern to, protect you. And to have the adequate airflow pattern, you have to have the shield has to be down a certain, where you can still put your hands in it and work with it. But if it's too high, then you lose the current of air. Fume hoods only remove harmful vapors, so like it wouldn't be appropriate to do like microbiology samples and stuff like that under a fume hood, because that's not its purpose. So you want to make sure that you use the right biological safety cabinet for the right purpose. So obviously for the biological safety cabinet that's installed in the microbiology lab is the appropriate safety cabinet, but you do have to operate it in the right way. So training is usually necessary for you to know what to do where. If sometimes you're trying to prevent yourself from getting contaminated by what's in the sample. But sometimes if you're doing things like PCR and DNA extractions and all that, you're trying to not bring outside contamination into the sample. So, there's different mechanics to all of this. And it's again, just important to follow protocol in the workplace. And then some of the other lab safety equipment are going to be all the needle stick prevention engineering controls, so the retractable syringe needles, the self-sheathing needles, sharps containers, etc. that we've already talked about. There are also fire suppression systems in the lab, eye stations, emergency showers, and pipetting aids and stuff, which you should never have to mount a pipette. It should be things like maybe these little fun little rolly things that you can use to control the liquid in and out of the pipette. And then a little bit on employee safety. What's the responsibility of the employee? Food and drink. So, for example, not take food and drink in the lab. So, same thing for you guys as students. We expect you to abide that no food and drink in the lab, and we don't have to be after you for that. So, that is your responsibility to abide by that rule, to not bring food and drink within the lab. To not mess around with hair, jewelry, and cosmetics. You're not, putting on your makeup in the lab, including lip balm and stuff like that. You're careful about having excessive jewelry. Things can get caught in the mechanics of machines and all that kind of stuff. So you usually want jewelry to a minimum. You want your hair up. Preferably no long hair that can dangle and get into samples or get trapped into machines and stuff like that. So if you have long hair, it needs to be up. And what's the responsibility of the employer or here the school is to provide you with eye and face protection and all the personal protective equipment that you require, including lab gloves and stuff like that. And so anything that you need for personal protection to work in the lab should be provided by the employer. All right, one more question. And so if you experience a bout of C. diff a day after you interacted with an infected patient, signs were posted and the isolation card was in front of the patient's room. So this exposure, is it your fault or your employer's fault? All right, good question. And the last one is the one that's going to be all the lessons. Just if you have any questions about this lesson, you're welcome to put it here. If you think about it later, you can just email me. So, all right, so this is your lesson for chapters 1 and 2, and thank you for your attention.